So thank you very much. My talk is not going to be very science intensive, so it's just a little bit of my who I am and what I'm doing here, but whatever. It's not going to be very hard to follow. Okay. So this is me essentially. I am actually a veterinarian that became a bioinformatician like eventually because they don't like the clinics so much. And then I did a PhD in Barcelona in investigating animal science and how muscle and fat metabolism works and how gene regulation works there. Then I eventually was hired in 2020, not a good year to start anything, but I was hired there in Sweden to work as a postdoc, to work a little bit on a mixture between aRNA biology and paleogenomics, so in paleotranscriptomics with Lowe Vidal and Mark Verlander. Then when those 2 years finished, I went a bit to work into for the Empire in Gothenburg in AstraZeneca. If you want to know what I did there, I cannot tell because I've signed up NDA. But I was not doing anything fancy, just some kind of RNA biology and stuff. And then I said, okay, I'm here in Gothenburg. I might want to go back to the capital in Sweden because I was living there with my boyfriend. And there I got this kind of position in the MBIS, which is the National Bioinformatics Service in Sweden, where I was essentially working with the same stuff. I was working doing transcriptomics and RNA-seq analysis for all the groups that PIs that were asking for help, to us. And eventually I wrote some grants here and there. I got this grant, the Billion Experiment one, and that is why I'm here, initially for 3 years. So yeah, this is basically me. So what is this all about? Why I'm gonna here? What I'm gonna do here? Why I'm here? Essentially, what I got this grant. So we all know more or less that this means to be like all samples tend to have no DNA or very bad DNA, but sometimes they have good DNA, and this can lead to a whole field being developed by just sequencing how this DNA works and how all those ancient animals and ancient plants or whatever were when they were alive back in time. So essentially, ancient DNA rules the whole thing. It's been a field established since the 80s, although in the past 15 years it's been developed through the NGS technologies throughout all the sequencing of the whole genomes of these all those animals like the mammoths, rhinoceros, Neanderthals, even macroorganisms. But what about ancient RNA? So why have we not heard about just these molecules? We just have heard about DNA and maybe some kind of proteins, but not very much about ancient RNA. So in essence, this is more or less how it looks like if you compare DNA with the case of the RNA. You can see the field of ancient DNA has been developed throughout all these years since the 80s. And it's now a kind of major field with lots of papers being out each day. Whereas if you look at the ancient DNA perspective, although there are a few more papers if you expand your view out towards other things that it's not animals, but there are just 4 or 5 if you more or less take the ones that are not very reliable. Maybe you can count 10, 12 papers out there so far. So the difference is quite big. So why is that? Why so few papers out there? Although we have had sequencing techniques since 15 years ago, and we have been working on that. Why so many papers on ancient DNA, but so few on ancient RNA? And maybe it's because it fails and it's not a real thing, or maybe it's because the people have not been working on it seriously. So one of the first issues is this kind of preconception that DNA is very, very harsh and very durable throughout time, and while while RNA is very bad. It's like okay, it's not gonna be durable. It's gonna be degraded throughout time. It's not gonna be there when you look at it. But our hypothesis is that, well, it might not be very strong, but it might be in certain occasions, and if some sort of conditions do happen to be there to be able for us to be able to go there and get those molecules and sequence them. So essentially, what I got this grant funded and this money for is okay. We have proved, and we have more or less some evidence that we can actually extract these molecules, these RNA molecules from those tissues, those samples that are out there in the museums, or that we get from the permafrost in the soils if we go there. But there need to be some certain conditions so that those molecules are going to be preserved through time there, essentially like similar to DNA cold desiccation, or maybe they have been conserved in ethanol or some kind of a formol thing, or maybe they have been preserved in these FFPE things, these tissues for the medical tissues. So in essence, what I aim to do is based on the assumption that we can actually recover those molecules, and they are still there and they have not been degraded over time. We can actually recover them, sequence them, analyze them, and gather some biological information that is actually relevant for us. And I aim to do it better and bigger because I've done it before, but just in a very reduced experiment when I was working in Sweden with Love Dahlia and Mark Freelander. If you want to know more about this, I have recently published this paper working on thylacines, where we gathered all this data in both muscles and skin tissues. And my aim now is to go bigger on this and to test if we can get better at doing this, because it's hard to do it. It is doable, but not very easy to do it. So if I'm new here, I know some people, but not many people, just a few of you. If you feel free to drop by, I am next to Tom's office. And again, friends, collabs, and random talking about life or science are always welcome. And I will be here for at least 2 years, or maybe more, if I get more money. But yeah, so thank you very much. This was all. I mean, if you are more curious about me, just ask me. I will be happy to talk with you. Yeah.